Okay, so this is a demo of biological databases, a follow-up to um, biological databases part one that we just finished. So we're going to look at UniProt. So a good way to start is to type UniProt on your Google's, and you will see the link to that. Okay, so you have a search bar on top. Once you get there, you can do just a simple search if you know the name of the protein, and just click search and you will get that. There are some other advanced search terms, in which case if you have a couple of terms that you want to combine for your search, that's also possible. I also mentioned the fact, and I can't remember if I did mention, that some of these databases also contain tools that you can use to analyze the information that you get out of them. You have the link to your BLAST. Um, I guess you can check out what BLAST means. It's not covered. I don't want to talk about blasting in this. Alignment of sequences is also possible. Retrieving the ID and mapping it, and some you can also search for peptide um, within this platform. All right. So I'm just going to choose a protein name. You can choose anyone you want um, in your. And I think I asked for you to choose specific protein in in the Anson tutorial. So I'm going to use a protein that I know is present in Plasmodium. I'm just going to say search. Okay. So it gives me uh, popular organisms where I can find it, and it gives me a list. So the entry here, the table here, that you get, because you have more than one uh, entry for that FAST13 protein. So there's quite a lot of entry for this one. About 69 entries here. So I can't pick one. I'm looking for the one expressed by PF3D2 3D7, this particular gene name. So I can click on that. It's from Plasmodium falciparum to 3D7 isolate. And it tells me the length of the sequence for that particular protein. So 413. Okay, if I click on that, now it takes me to the page for that particular protein. And so we start by looking at the protein name, FAST13, the gene ID for that protein, the organism in which that protein is found, Plasmodium, and the status. Now, like I said, once you put in a particular information, people are allowed to upload data into some of these databases. However, they go through process of reviewing the data that have been deposited, and they kind of annotate that. So you see here, it said it's unreviewed. It means that they are still reviewing that. Once they are able to complete the review for this, the annotation score will be high. It's a predicted protein, predicted to be present in that particular organism. We can check out more information on the review state. Okay. Now, there are major areas. The major areas in UniProt are actually on the tab here by the side. So you have the function, the names and taxonomy, the subcellular location for that protein, the expression, and the processing, molecular processing for that particular protein. In terms of the signal peptide in that protein, and the proteomic databases, especially for those that do proteomic experiments, the structure of that protein, and the family and the domains of that protein. Now, all these are linked to specific databases that actually gives you information on these different aspects uh, that are outlined here. Similar protein also, so this is the sequence of the protein. It also talks about similar protein and references. All right. So if we go back here, you will see the go time for that particular protein that gives you indication or as to the function of the protein. So by go time, I'm making a gene ontology um, term for that. So you can actually go to the gene ontology page to see. So it's a cysteine type endopeptidase inhibitor. That's the activity of that particular protein. It's involved regulates the entry of the parasite into the host, particularly the erythrocyte cells. Okay, that's what the biological process is. You can also click on that to get more information, or you go to the complete annotation on Quigo. All right. I can't cover annotation here, but yeah, you can go to the gene ontology and annotation to really get more information about that particular gene or particular protein. All right. So in terms of names and taxonomy, it gives you the name. The gene name is written here. There's a link to that. This is the organism. Taxonomy identifier. You can get the phylogeny information here. So I showed you the VEUPATH database. Okay, see, there's another link to that database because you have information about this in full information about the genome of the Plasmodium in this particular database. Then the subcellular location of this protein in the cytosol, in the host cell cytoplasm. Yeah. The domain of the protein and the sequence of the protein. So what is what we call a FASTA sequence, which you can download. The FASTA sequence. As a matter of fact, if you click on it, it gives you this document. You can actually copy this and just put it on in a text file and save it as FASTA for you to do some other things. All right, cool. But what I actually want to, there's still similar proteins and the references. But what I want to focus on, based on what we're going to do, is the 3D structure. So usually, if the structure of this has been resolved, you will have a PDB ID for this particular protein. So this is basically what I'm saying is this. So if I go to another UniProt site and I type in, I'm trying to find the protein. Okay, I'm just going to call it ACE2, which is your angiotensin converting enzyme 2, which is the receptor for coronavirus. In the human, if I click on that, so angiotensin converting enzyme 2, ACE2, gene name, present in Homo sapien human, and applies a function here. But this is where I'm going in terms of, so if you go to this tab, you can actually click the tab and it takes you straight to the place, the structure. Okay, so this is what you see. This is actually the structure of this particular protein. So you don't have that here because if you go to the structure, you don't have it here because the structure is not available in the protein data bank. So in this case, so many people have worked on this protein and deposited the structure in the protein data bank. Okay. So you can actually go to any link from here to actually go and see the structure. And you see, like I said, the databases also provide some tools for you. They have a visualization tools here for you to look at that particular protein. All right. But for if you don't have that structure in Protein Data Bank, there's some other databases that, what's the word I'm looking for, that try to model the structure. So you have Modbase, which is a site that tries to store. Some people have said, okay, fine, we can, based on similarity to other protein, model the structure of this particular protein, even though we have not experimentally established the structure. We can build a model from the knowledge we have from other databases. So you can have that where somebody has actually build a model. This is what you have. Somebody went to build a model for that particular protein. Yeah, and this is what it looks like. Okay. So you if you really need to work on this, you're not limited. You can actually go and get um, the structure from that particular site and begin to work with what you have, rather than saying, okay, there's no structure, and that's not a limitation. The other option will be to, if you go back to the sequence, which is here, you can take the sequence, download the first file, and take it to some other platforms that can actually build the model based on the sequence of that particular uh, protein. Okay, so that is also very possible. All right. So just once remember, you will definitely have some that have structures in Protein Data Bank will be shown here. Yeah, and you can select any one of them you want. Look at the secondary structure is also shown here, applying the helix, the turn, the beta strand, yeah, of the protein. All right. So I'm going to, yeah, so you're going to play around with UniProt, and you're going to use it to get some things. Okay. So let me stop there for now. Then we'll go to Protein Data Bank, which I think is more of what we want.